right, so our second part of genetics, we're going to finish the example. And when we when we did the first screencast, we said that Herman has no widow's peak. Notice no widow's peak here. So that means that he has two recessive genes for widow's peak. So he has two lowercase w's to represent those two recessive genes. That means that he is homozygous. Homozygous again means same genes, and he has recessive genes. So same recessive genes. Lily, however, could have two possibilities for her genes. Because she has a widow's peak, there's two different combinations that could give somebody a widow's peak. The first combination is she could have a dominant and a recessive gene and be heterozygous for the widow's peak. She's still going to have a widow's peak because if you even have one dominant allele, you're going to have and show that particular trait. But also she could have two dominants. That would also make her have a widow's peak. She would then be homozygous or same dominant, so she would have the same two dominant genes. So our next job is to try to figure out which of those two combinations does Lily have. So we are going to cross Lily and Herman's genes using a Punnett square. So we said on the first screencast for genetics that a Punnett square is just this little tool that can help you determine the offspring possibilities for two different parents. So when you do a Punnett square, it's just 4 boxes. You're going to put one of the parents on one side, their two genes, their two alleles, and the other parent on the other side. And it's not going to make any difference which parent you put on which side. You're still going to get the same results inside the Punnett square. So let's imagine if Lily is heterozygous, meaning again that she has one dominant gene and she has one recessive gene. You just write the letters that represent her two alleles here at the top of the Punnett square. Herman, we already know, is homozygous recessive. He has two matching recessive genes. So we're going to put small w's on this side for Herman's genes, Herman's alleles. And then what you're going to do is you're just going to bring this dominant one down here and this recessive one across. So that's going to be a dominant and a recessive. The dominant gene drops down, the recessive across, and you end up with the dominant and recessive in this box. Here we have a recessive and a recessive for two recessive alleles. And then same thing, bring this trait down, this allele across, and you end up with two recessive alleles. So this shows us the offspring for Herman and Lily, if Lily is heterozygous. Okay. So notice that when you cross Lily with Herman, we're going to get 50% of the offspring will have a widow's peak. This is dominant. Even if there's only one dominant allele, that's the trait that you're going to see in the offspring. However, they also have a 50-50 chance of having an offspring that doesn't have a widow's peak, because here we have two recessive alleles for widow's peak. So this is going to be a 2 out of 4 chance of a widow's peak, or 50%, and a 2 out of 4 chance of having an offspring without a widow's peak, or again 50%. But we said there was another possibility for Lily. So we have the possibility that she could have two dominant genes for her widow's peak. And again, we're just going to put those into the Punnett square. So we're going to put her two dominant alleles here, Herman's two recessive alleles here. We're going to bring them down and across to fill that square, down and across here, down and across. And notice here, if these are her alleles, that all of the offspring are going to be heterozygous. They don't have a matching set, and they are going to have a widow's peak because they have one of the dominant genes for widow's peak. So now we have to decide and figure out which one is true. Is Lily homozygous dominant? Are these her alleles that give her a widow's peak? Or are these her alleles that are heterozygous that give her a widow's peak? Well, in order to do that, we're going to have to look at the children. Here we're going to look at their offspring. This is going to give us a 1 out of 2 or a 50% chance, 2 to 4 ratio of having a widow's peak. Again, half would have no widow's peak. If she's homozygous, if she has two dominants, then 100% of their children, 4 out of 4, would end up with a widow's peak. When we look at the other children, if you ever watched the show, you might remember that Herman and Lily had another offspring named Marilyn. This is Marilyn. And Marilyn has no widow's peak. So by looking at the other children, by looking at Marilyn, we know that this has to be Lily's genes. She has to have a recessive gene so that she could have passed those on to Marilyn. So this is Lily's alleles and not this. She cannot, if these were her genes, she could not have an offspring that had no widow's peak. All right, so the Punnett square is just a good way to look at what type of offspring you get from crossing two different parents. So you put the females here. This female happens to have a dominant and a recessive. The male has a dominant and recessive, and you're just going to, for each box, you're going to bring the allele over and down. So you can see here in this example that they brought the recessive allele over, the recessive allele down, and so this offspring would have the probability of having two recessive genes. Now, one other term that you're going to need to know is, I guess two other terms, is genotypes and phenotypes. Genotypes are the genes that the organism has. So right here, these are the gene possibilities for these particular pea plants. The genotypes are the genes. They are represented by the letters. So if you're asked for the genotype, you're going to give the genes or the letters that that organism has, because the letters represent the genes. The phenotypes are what you see physically in the organism. So think pheno starts with PH, physical starts with PH. So that's what you see. So you can see here in this example, if a pea plant has two dominant alleles, and that's its genes or its genotype, you're going to get a yellow phenotype. That's what you're going to see physically in the appearance of that organism. Here, this genotype, a dominant and a recessive, is going to give you a yellow phenotype. And two recessive genes in the genotype is going to give you a green phenotype. So you have 3 different possibilities of gene combinations, but only 2 different possibilities for how it looks in that particular organism. Remember, genotype are the genes, the letters, and phenotype is the physical appearance. What do those genes look like in that particular organism?